ask my co-chair, Dr. Chandramani Pathak, to introduce the subsequent speaker.
Thank you, Dr. Padmalini. So our next speaker is Dr. Girdhari Lal. So this my great pleasure to introduce very eminent and young scientist, Dr. Girdhari Lal. Dr. Girdhari Lal is senior scientist at NCCS Pune, and Dr. Lal received PhD degree from Indian Institute of Science, Bangalore. He was assistant professor at Mount Sinai School of Medicine, New York, USA, before returning in India year 2010. And Dr. Lal is interested in understanding the cellular and molecular mechanism of immune tolerance. His laboratory is investigating the role of chemokine receptor signaling and neuroimmune communication in inflammation and autoimmunity. He has published more than 50 research articles and presented his work about 150 national and international meetings. He received several prestigious awards, including Ramlinga Swami Fellowship, Innovative Young Biotechnology Award from Department of Biotechnology, and Swarna Jayanti Fellowship from Department of Science and Technology. So Dr. Lal will deliver the talk, Cell Receptor Cellular Communication and Linked Disease. So I invite Dr. Girdhari Lal to deliver the talk. So
thank you, Dr. Pathak.
Dr. Girdhari Lal.
Yeah, I'll just share my slide first. This slide is visible?
Yes, it is visible. Yeah.
Perfect. So let me start with thanking the Society of Inflammation Research for putting this program together, and also the audiovisual team, which is now communicating all this, what we can share now, the information. And I was given task on speaking to cell receptors, cellular communication, and linked diseases. I think most of these things are covered, and we saw that in the previous talks that there is a complexity in the immune system. And there are a lot of molecules and receptors involved in it to either communicate the message from one cell to other or to the within the cells itself. So what I will simplify quickly is that if you look in a crowd, just like immune cells, if you look in the crowd, you will have different caps, or what we can call it as here in terms of that you have a different receptor. And if we have to call them, each one of them, then it will be like everybody will respond, probably possible or not possible. But if we segregate this, that okay, those who has red cap are versus green, or versus something else, I think then group of people will respond, group of people will still thinking then what to do. And likewise, what we have in radio signal, that all the radio waves are around us, but we don't listen when turn on the radio until unless we right tune to the particular signal, particular place station. So what it means is that in immune cell also, we have a lot of cytokines, lot of molecules around, but does all of them are responding all the time? Probably not. So how they decide? Depending on what they have on their surface, like here I said, and they based on that, they decide that what to respond and how to communicate to each other, either positively or negatively, what's going on in their surrounding. Now, message, what we want to say here is just like you have communication to person who is very, very naive, like our immune system, naive immune cells, or person communication to only a specific set of cells or tissues, or to the cells in the tissues, as like a group communication to a specific and targeted one. Or you can also have communication going on to the remotely and still understanding what's going on around us. And of course, self-communication or signalings within what's going on in the surrounding. All right, so that's just the thing that how cellular communication goes on in our body. It was Dr. Swaminathan also described it, that the cellular communication, what happened is that in case of this, you have endocrine signaling, and then you have parakine signaling, contact dependent signaling, and all these things that either signaling coming from very close proximity or traveling through all the way from one location to other, or within the different two cell types through a contact dependent manner. So in all these terms, what you require is a molecules which receive the signal and a molecule which provides the signal, and then the communication happens between two cells, either close by or the far away. All right, so now if you look at molecular terms in terms of communication, that you have different types of cell types involved, and the molecules either coming from extracellular environment, it can be pathogens, it can be any other molecules in the surrounding, or within the two different cell types through receptors, they can communicate each other. And you can see in the real life situation that yes, cells do communicate through a contact dependent manner through a different pathways or different vesicular format. And that's how the different languages are used by cells to communicate. So when I say languages of communication, it can be just what we are saying as by touch or by coming in touch of molecule produced by them. So there are a variety of ways the cell communicate in the body. We will restrict ourselves today as immunology discussion, that when we see in clinic or in terms of inflammation, what we see is that a specific tissue, mostly some of the autoimmune diseases targeted to a specific tissue. Now, what kind of communication these particular organs, either in gut or in the skin or in the lung or in the brain, wherever it goes, what kind of communication immune cell receive, so that the disease what you see only in a specific targeted organ, and how these immune cells communicate each other, so that what you see that is not a systemic, not all the time systemic disease, although traveling through all the way systemic pathways, but targeted to a specific tissue or organs. So now what we will discuss here, that how specific communications between the cells are arises, how they communicate to each other, to the close by and to the far away distances, and the speed of communication, that what's going on around in the surrounding. All right, so when you have to communicate, you need to have a postal code and postal address, that where to go and how to go. So where to go means they have to have the address, and these are some of the molecules which is present on different tissues as a receptor on different tissue, which guide the mole cells, which are expressing a kind of receptor which actually direct them either to skin, lung, gut, liver, or peripheral lymphoid tissue or secondary lymphoid organs. So now you have to have two type of things. Somebody should invite you. So that means you should if the immune cell want to go, they must know where to go, and that is guided by different kind of cell adhesion molecule or chemokine. Like if you have the expression of CCL1, 25, 2, and 5, then corresponding receptor express on immune cells, they direct them to the skin. So as if you have corresponding cognate receptor and ligand interaction, it will go to lung or in the gut or in the liver. So now what you require is that a specific molecule, a address code, and a person which is going, or the cells which is going, they should express right receptor to reach to the place. Otherwise, they will be just floating in circulation. All right. So now when we say the lymphocyte recirculation, then it's the route. And in time and space, when cells comes out from the bone marrow or in the circulation, you will see that mostly cells will stay a specific time in the different organs before they go into the different tissues itself. So if you look into a spleen, mostly the cells comes in circulation, at least minimum 5 hours or close to 5 hours, they spend time in the spleen. Up to 40%, 45% of the cells which in circulation, and 42% of cells which actually circulating spend almost 12 hours into the lymph node, and finally they distribute into another organ. So now when they distribute and come in and go, there are specific pathways, there is a specific route which they follow. And in the lymph node, which I'm showing here, that cells, when they come in circulation, they go to the lymph node and the lymphatics, and through efferent lymphatics, they can actually enter in the lymph node. Now, when they enter into the lymph node, they actually travel through a specifically designed path or designed structure where they interact with a lot of immune cells, and then finally end up with the going outside to the outside world. So whatever happened here is the required signal to come in and the required signal to go out. And all these things are guided by different molecules. So what you are seeing is high endothelial granule that provides entry point for immune cells to go in, who can go in or who cannot go in. And by this way, the cells actually travels. Now, what I'm showing you here is again the lymph node structure, but here what you see that the lymph node structure is very organized structure. Now you see that in a place where cells actually comes in through efferent lymphatics, they have to encounter the B cells, naive cells here, and then once they interact with them, they get training, and then they exit to efferent vessel, lymphatic vessels outside. So interaction of this kind, and there are underbase cells directly which in circulation can come in, and these cells actually uses the particular guarded gate, which is high endothelial granule, and through this they can come in, but this is restricted for specific cells. So there is a specific requirement who can go through this route. There are a specific requirement who can come in through this route, and there is invitation or postal code which decide who can come here or who can come here or who can come here. So if their address is to only this zone, that is T cell zone, they can stay up to here in this boundary. If they have address to only this location, they can go. Or if they reach there and then go here. So it's a interplay of expression of different chemokine receptor, and cell adhesion molecule, which require them to go to different location. And this is what you see. The cells are actually well behaved, very organized structure, where you see that dendritic cells are in a specific location. Then you have B cells in blue here, and the red cell, the red cells and the blue cell in the. So B cell is here written in the blue. They actually are in a specific location. So it's a well-designed structure, and cell must come into a specific way. If there is disturbance in any of these movement or pathway, or even disruption of this can lead to different kind of inflammation or autoimmune diseases, or even not mounting the appropriate immune response. I'll take example here. So now the cells which comes in through the efferent lymphatics, actually the reason for them to come, if you see this B cell zone, what I was showing to you, so antigen captured dendritic cells comes in, and then they get exposed to the B cells, or even T cells using a different mechanism. Now, why this is important? Why this structure? Why these receptors are important? I can give you one example. Most of the time when we do experiment in the Petri dish, and let's say we have some antigen which in vitro stimulate one cell type which secretes cytokine of whatever type you're looking at. You're looking at inflammatory or anti-inflammatory. When the same antigen is given inside the body, it doesn't respond. So what you have in Petri dish in open structure, where there's no barrier, where there's no structure, organized structure, the response what you see is different. And because I showed to you that there's a guard, there's a specific requirement who can come in, and this specific requirement is very, very important for mounting effective immune response. Since now people what you think that they have the molecules which generates a lot of inflammation or a lot of anti-inflammatory molecules, let's give it, and then this might actually generate immune response of their choice. It doesn't happen. So because these guards are protecting them not to come in, just like example of nanoparticle. People see that lot of nanoparticle coated antigen, very good in Petri dish, but not in vivo. So because the trafficking of those molecules, if not in this way, they cannot mount immune response. And to address that, if you inject directly to this location that is specific nanoparticle or antigen, you start seeing those immune response generating in the body. So what it means is that when you have this type of organized movement and organized way of cells to traffic to the different location, you actually contribute in immunity, mounting. I'll give you another example here. So a lot of time what we see is that immunotherapy, a lot of antibody is used, and immunotherapy in terms of checkpoint inhibitor, now everybody is aware of it. I'll tell you one example where people has done and shown that these structures are still important for those checkpoint inhibitor to work as anti-cancer therapy. Example here is given in a mouse model where MC adenocarcinoma cell line was used. So if you take mice and if you don't do, just inject these tumor cells, tumor keep on growing. And if you so in this experiment, what was done that the lymph node was removed that is close to the where the tumor cell was injected. Now, if in the mice you have tumor growing, and in case if you give PD-1, then what will happen? The tumor actually, because of the nature of the checkpoint nature, it's actually promote the tumor anti-tumor immunity, and tumor stop growing. Now, in this case, if you just remove the lymph node and you still give the same molecules, now no longer start working. Actually, the anti-tumor effect, what you see within these molecules lost. What it suggests that the same thing is presented here as a survival data, that if you actually disturb the lymph node, you don't actually get the activity what you're supposed to get in presence of lymph node. So organized structure is very, very important. Now, when cells talk to each other, cells communicate each other in different ways. So like here, what is shown as a dendritic cell communicating to T cells, and in the T cells, the part or the region where the synapse formation or the contact point, there are specific molecules and receptor come into place. And now these are called as supramolecular activation cluster at the synapse site. What are those molecules? The molecules which is in the eye, you can see very close to the central point, which is called as central supramolecular activation cluster. There are set of molecules like T cell receptor, CD4, CD28, they participate in forming this central communication point between T cell and antigen presenting cell. There are cell molecules like cell adhesion molecule, LFA1 and ICAM1. They actually participate in the peripheral system. There are other molecules which are distally located or DSMAC, or so these type of molecules actually do communicate. The interaction of these molecules play important role even for T cell or APC T cell interaction in mounting immune response. So there are a variety of receptors which participate in, and some of them in a specific way they have to communicate in order to induce a proper activation signal between the cells. All right, when cells also travel from one place to other, they have to move on to one location to other. And when in circulation, they decide to go to other place, some of these molecules actually start playing them either in terms of stopping their rolling or arresting them. And then the cells decide using a specific molecule, this is the right place to go in. So in different process, all these cells require different molecules. And if we have trouble in any of these molecules, then we'll have trouble in terms of generating or mounting the immune response. So in terms of molecules, you have the cell adhesion molecules, and these are some of the molecules like selectins, integrins, and number of integrins are involved. Our immunoglobulin superfamily cell adhesion molecule are CAMs, which are different molecules. And I told you that all these molecules are different location and different place within the synapse. Actually, they contribute, and also they contribute in the migration. Now I'll take the example of here that there's a problem in cell adhesion molecule that is called as leukocyte adhesion deficiency LAD syndrome. What happened in this case, that if the molecule like integrin, which participate in the movement, if they have a trouble, they actually create, the cells actually had problem in either rolling or activation or adhesion. So leukocyte adhesion deficiency, different grade of this particular syndrome from 1, 2, and 3, and different molecules are participating in these diseases. So fine, so when you have trouble with these receptors, the interaction of these molecules, and then you end up with the one or other diseases. Other molecules, very important molecules, is a chemokine which guide them. Other than these postal address, they know how to go about. If so, this looks very complex. There are chemokine receptors. They are named as the CC chemokine, or they have been also named as CXC chemokine. The two cysteine residue separated by one amino acid, or two cysteine residue separated by 3 other amino acids. So that's how they named as CX3CR receptor. And correspondingly, the ligand which binds to it, they have been known as the CX3CL1. Now there are one chemokine receptor, and there are several chemokine ligand which can bind to it, and there's the n number of ligand this bind to it. What does it mean? This is confusing. I'm pretty sure nature has not designed this way to confuse the system. Now, what we need to learn from here is that the receptor, which is now directed by different chemokine, we need to understand that who should express the receptor, what cell type it express, and which chemokine is produced by which one of them, which cell type and which location, and how these cells are guided. I think there are a lot more things need to learn where there is a redundancy in the system. Probably it's redundancy because we don't understand, but if we understand carefully that why nature has created this redundancy, I think it's an important aspect to learn. There are chemokines which has only one ligand, like here CCR9, CCL25, it has only one ligand and one receptor. There are like CCR6, these. So there are some specific chemokine and chemokine receptor pairs which are present, but there are redundant system. And now we should understand that biology of this redundancy. And it's not a nonsense. It's probably nature has created for a specific purpose. I think we need to learn from the nature why and how, what is the purpose of this. Now, again, what I mentioned to you that how cells decide from one place, let's say circulation, to go in bone marrow, thymus, or spleen, or lymph node, or skin, or any other part of the body. So now you see that the chemokine expression pattern, the receptor for chemokine receptor expression pattern on a specific cell type actually changes when they are traveling from one tissue to other. So now this is very important that who expresses a specific receptor, where this specific ligand is interacted. Now, what is the combination of these two receptors and ligands which guide them to go, even though the cells express, most majority of the cells express some kind of chemokine receptor. So now it's a very complex biology, but what I want to show from here that still the cells are guided through this chemokine receptor to go into a specific location. And sometimes we qualify some of those cells which are expressing the specific receptor of a specific subset type of, let's say we're talking about T cells, then we can call it as a specific subset of T cell which express a well-defined or majority of the time specific chemokine. Now, how does these chemokine receptors are altered? There are expression of pattern recognition system or TLR molecules, which actually alter the ligand expression in specific tubes, or some other cytokine alters the ligand expression, and which directs the cells or recruit the cell to the site of inflammation. So there is a variety of receptors which help in the communicating to the cells where to go, which location to go, using the circulatory system, either blood or lymphatic system in the body. Another example is CCR6, which I showed to you that it has got only one ligand, which is CCL20. And if you look into the disease, a lot of disease are having association of in the contribution of pathogenesis with the expression pattern of CCR6. And if you knock down this receptor or reduce the expression of this ligand in a specific tissue by using antibody or any other intervention mechanism, you see majority of these diseases are subsides or controlled. The severity of the disease is controlled. So now it's suggesting that depending on the nature and the tissue which chemokine is expressing, it actually guides the cells to go in a specific tissue. It can be mostly pathogenic or even under homeostatic condition, some cytokine. I'll take another example, which is CCL9. It's another CCL9, CCL25 and CCR9. Now, CCR9, when in the birth, in early stage of life, it expressed only, CCL20 only expressed in the thymus. Now, as we grow, the expression of CCL25 direct from thymus to the gut. And now, once the expression of CCL25 initiate in the gut, the cells which are expressing here, the CCR9, they start contributing or mobilizing to the gut. So now CCL25, mostly the cells, epithelial cell, thymic epithelial cells express this. Now gut epithelial cells start expressing. Now you have the cells or molecule at different stage of life that they are contributing the mobilization of the specific cell. And now what we have shown and what is known is that the cells which are expressing this, depending on which cell type or ligand is interacting, it can modify the phenotype of those cells. Like here, if dendritic cells are exposed to CCL25 in the gut, they actually through lymphopoietic stromal cells, they promote Treg cells and suppress the TS17 cell. And likewise, there are other pathways which we need to understand that how these chemokines communicate to different other cell type in terms of contributing the either immunoglobulin production in the gut or in different location or in the inflammation. I think a lot more things need to be learned where the cell receptor, which is known to help them to migrate, but also contribute in the intrinsic signaling in the differentiation and controlling the function of two different cell types like inflammatory and anti-inflammatory cell types. All right. When we look at disease, I'm not listing here all. There are several chemokine receptor has been tried out in clinical importance. Some of them are listed here like WHIM syndrome, which referred to as warts, hypoglobulinemia, infection, myelocathesis. In this case, what happened is the gain of function mutation in the CCR5. And in this disease, you will see the kids will start developing warts in the hand. Now, how do you identify when they actually start antagonizing these receptors? They see that there's a control on this. Likewise, other receptors, CCR5, which is, we know that one of the co-receptor for HIV infection, and there are drugs which target them. And these molecules also require for the migration of the cells to different locations, especially in the skin, out in the peripheral tissues. Atherosclerosis, we know that another type of inflammation where some of these chemokines do participate in, and people are trying to target these by targeting these chemokine in the atherosclerosis. Sometimes when the antibodies form against this autoantibody, which in heparin-induced thrombocytopenia, they also contribute in autoimmune diseases when there's antibody forms against this. All right, so now what actually we heard in the morning that Dr. Subramanian, it was told that Swaminathan has told that that cell, depending on nature, it can be inflammatory or the immunosuppressive. So that's correct. It's actually that's what one example which I want to give here is IL-2. The receptor for this expressed by different cell types. And mostly it's inflammatory cytokine, it was tagged. And in an example which I want to put here is that in cancer, what happened in cancer, there is immunosuppression. So what people thought, because it's an inflammatory cytokine, in the cancer patient, if you give IL-2, maybe there is more inflammation generated and then it will protect. But when they did the treatment with IL-2, instead of suppression of the regression of tumor growth, tumor started growing even much bigger. So what we understand in the beginning that it has immunostimulatory role, which suggests that now it also participating in anti-inflammatory pathways. And the way it does, and later it was discovered that IL-2 also promote secretion of IL-4 in the Th2 cell, and also induce apoptosis. And most importantly, IL-2 is also required for regulatory T cell, which is a suppressor T cells. Now, if IL-2, if you pump more in the body, the Tregs goes up, and in a cancer patient, you really don't want this cell to up. So that's why when we learned that IL-2 has both, then people started realizing that yes, it's not going, and then we found the answer that why IL-2 in the patient, which was known as activating molecule, now it's suppressing the immune system. Another molecule is TNF. TNF also has got dual role depending on which cell type you're referring and which tissue you're referring to. It can have immunostimulatory and immunosuppressive. Now TNF, how does it do? It depends on what receptor it is binding. Sometimes when it interacts with TNF receptor 1, it has got different function. And when it interacts with TNF receptor 2, it actually got antagonistic function. Depending on now which cell type TNF is binding, where actually in which tissue you are referring to, it may have both type of functions. So the time, the type, and the location of this cytokine is going to decide what phenotype is going to acquire as an immunostimulatory or immunosuppressive phenotype. All right, now come back to another location where central nervous system, again, communication to the brain, and communication of inside the brain to the immune system is really tightly regulated. But if you look at the vascularization of the brain, you will find that a big vascularization is inside the brain. But still, immune cells or most of immune cells do not have access to the neuronal tissue. And in terms of number, around 2% of body mass brain is representing, and 20% of your body's glucose is and oxygen is used by the brain only. In terms of how these tight regulations are maintained, and how does any cell type during strong inflammation decide to cross this blood-brain barriers? So we and others have shown that under certain circumstances, the barrier which are always intact because the cell adhesion molecules, the cadherin and molecules between the endothelial cells which forming these tight junctions and form a very controlled environment under influence of inflammation, these tight junction molecule or cell adhesion molecules can be damaged. And once it is damaged, the immune cells can go in. And when they express the neuronal tissue, that's where we have CNS diseases coming in, starting. Now, the cytokine which is produced here and the cytokine which is required here, they can be different. So the molecules which damage this barrier, and when cells go inside, the molecule which require for pathogenesis in the brain, they might be different. I think we need to understand more details in the biology of this. Who breaks the barrier and who creates problem inside? All right, very good. So now what we saw that in the yesterday and today's talk, that you require a multifactorial and multidimensional interaction of different system exist in our body. And you just disrupt one of these system, the balance of these two also get disturbed, and then we'll have problem in our body. So either you induce hyperimmune response, then you have your nervous system goes differently. And if endocrine also disturbed, these two also behave differently, and then there is a loss of homeostatic balance. So now how these cells communicate, how the immune cells and nervous cells and the cells which is endocrine system, how they communicate to each other. Now let's talk about one example or one tissue where is the gut, the one organ which is gut, we call the second brain. A reason for calling it is second brain because a lot of nerves actually end here. And the gastrointestinal tract is very big, 5 meters in length. And if you just open all this and make it a single seat, and it is almost 22 meters square meter surface area. And with this much surface area, with lots of microbes inside and controlling the immune system to that level, keeping the balance between the disease and the healthy condition, there are a lot of neurons also draining and which is sensing all the time. So the immune system, how they actually contribute, where the lot of our immune systems are there, how the neurons are communicating in the brain. And that's very interesting area to learn. Anything disturbance between this, we actually see we end up with the disease condition. So how does neurons actually sense immunological molecules, and then they talk to each other, neurons and immune cells? So if you see carefully that the neurons express some receptors, which are mostly known in immunological point of view, like interleukin-1 beta, TNF receptor, IL-6 receptor. These receptors are like cytokine receptors are mostly known in the immune cells. But now you see that these receptors are also expressed on neurons. So when there's a cytokine produced, it also is not activating only these cells, they also give signal to the neurons. And that sends the signals to the central nervous system. And if you look other way around, the molecules which is mostly required in the neurons, now the receptor for these molecules are also expressed by some of these immune cells. So when there is a firing of some signal from neuron, and if there is a leakiness at certain places, or if secreted in the microenvironment, they also modulate the function of these immune cells. So there is a communication between nerves, and there is a communication between nerves and immune cells, and that's how they work out a strategy, how to maintain the homeostasis. If any disbalances happen, and that contribute in the disease or in inflammation. I'll take one example to show that, like you see that in the motor reflex neurons, you have neurons connected each other. Finally, when the neurons, one signal start from one place, goes to signal other place in the muscle to withdraw for whatever nosebleed signals you receive. Likewise, inflammatory reflex, so you have the neurons which stimulates the cytokine signals. Neuron senses pass on to distal location to distal organs where they actually produce neurotransmitter, and these neurotransmitter also is accessed by the immune cells. Now the function of these immune cells can be modulated by signaling through from one location to other and communicating between the neuron. I'll take one example to justify this. What was done in this experiment is that you take mice and just perforate or cut the intestine. So now you expose the gut microflora in the intestine. What will happen? Because of sepsis, mice will start dying. So now the survival will with time goes like this. Then experiment what is done here, you take the mice, but the nerve, vagus nerve which connects the spleen, just disconnect it by surgical disconnection and do the same perforation. Now you'll see the mice actually dies even faster. So when you disturb this secondary lymphoid organ communication and nerve communication and the spleen, the survival of animal through the sepsis is much faster, much low actually. The same way you can do the experiment, you electrically stimulate these neurons which are draining to the spleen, and do the same experiment. Now survival is improved. So now there's a communication between the nerves and immune cells, which actually contributing in the inhibiting the sepsis or strong inflammation. You can do the same experiment with, instead of electrical stimulation, you can do with the chemical agonist or agonist, and but you will see the same increase in survival of the animal. What it suggests that there's a neuroimmune communication, and this communication plays very important role. And I think now people are trying, there are clinical trial going on that how to, in arthritis patient, how actually just by stimulating the nerves, can you control the inflammation in the joint. So that's a clinical trial. There are success, there are hopes. And I think that this type of neuroimmune communication will help understanding more biology, interesting area of biology in the pathogenesis of disease, and also controlling the disease. All right, so before I stop, I'll just say a few words about the place where I work, National Center for Cell Science. It's a department of biotechnology, the institute, and you all are welcome to visit us. Of course, let the COVID go down. Hopefully, we'll have some meeting together in the NCCS rather than the virtual sometime. And NCCS obviously working in different areas of modern biology, including cancer, stem cell research, which you heard just now, then immunology and microbial ecology. And I stop here. Thank you all for listening, and I will take the questions.
Yeah, it was a very interesting talk, Dr. Giri. Does this receptors do explain the influence of psychoneuroimmunology? How does mind influences the outcome of some of the disease?
Right. So very important question, and I can tell you that people have done study on that. That if you see the people who are highly depressed, and they have more inflammatory cytokine in their serum. There are studies, and then if you intervene some of these inflammatory cytokine, you see there's an increase in their behavior, whatever psychological behavior. So there is actually some study where they point out that yes, some neurological and immunological communication do exist. Yes.
I can add a point to Dr. Girdhari Lal's response. So one such communication is the dopamine. So there is a very active dopaminergic receptors. I'm not talking about neurons. I'm talking about the receptors and the dopamine production and the inflammatory. We should not be calling inflammatory anymore. So cytokines effect on dopamine receptors on the neurons actually has this clinical impact or physiological impact. Yeah, it's proven.
Thank you. Right. Yeah. Thank you.
So thank you, Dr.